In this module, we will talk about the foam control system in fermentation process or in a fermenter while talking about the fermenter design. Foam control is an essential element of the operation of a sparged bioreactor. So in case of the mixing, agitation system and oxygen delivery system associate to each other when they work and the process is going on, then what happens? Then depending upon the nature of the medium, what happens? The foam will produce. As you can see in this picture, that the head space is totally filled by the foam. This is just an example of 2-liter laboratory reactor. So, if in such case, when there is a condition when foam is produced, the production of foam can affect on the oxygen solubility on one point, and the second, the existence of the foam that also make the column, and then there is a loss of the fermentation medium to the exit air point. So the excessive foam formation, when that tend to go from the exit point, that can choke the air sterilization system, which we called as the filter. So in such case, then there is any disturbance. So that can cause any pressure inside that. So there are so many happening that can happen. So in later case, we can do that. We use, when there is no blockage, either there is a removal of that, and when we have no any direct manual lock-in, what happens? Then there is a gradually loss of the medium, and then we cannot have some, in case in the absence of the operating personals, then there is a risk. So to avoid this risk, in case of the foam, then the foam has to be controlled. The foam can be controlled by the addition of some chemicals. So the chemicals having a two type of nature. One is antifoaming properties and other has the defoamer properties. So some chemicals known as the antifoams mean they hinder the production of the foam. So we add some antifoaming agents before the formation of the foam. So when there is a foam produced, and then we can add some defoamers. But commonly the word is antifoam is used. So if we add a suitable antifoam, but there is very critical while having selecting and the choice of the antifoams. In later module, we will talk about that what will be the quality of the antifoams, because antifoam has to directly enter into the fermentation medium. That can have the toxic effect on the organism, as well as mostly the antifoams can also affect on the oxygen solubility. So that's why we have to be very careful about selecting the antifoams, which we'll talk about in our later module.